This is a production of Cornell University.
So thank you so much, Dan, for that introduction, and thank you everyone for coming out to the seminar today. I'm really excited to talk with you about my work looking at the influence of microbial community dynamics on the cycling and stabilization of soil carbon. So the first piece of this story kind of comes into why are we interested in soil carbon? So where that comes in is looking at how soil carbon or soil organic matter relates to this concept of soil health. And so soil health is this idea that we have our soils maintaining this continued ability to be a living ecosystem that can sustain our plants, animals, and humans. So in agriculture, looking at maintaining a sustainable and productive soil system. And where soil organic matter comes into this equation is that we see that it contributes to better soil structure, it can modulate soil water availability, it's an important store of soil nutrients, and then also looking at the global scale, it's a globally important part. So especially understanding climate change, soil organic matter is an important part of that. So if we have this goal of increasing soil health, we also see that increasing soil organic matter can help us achieve that, particularly in our agricultural soils. And so if we want to have this and achieve this goal, we need to understand how soil organic matter is accumulating and also how it's retained. And so some of the questions we're asking ourselves when trying to approach this are what compounds make up soil organic matter and where are those compounds coming from? So last week we actually heard a talk from the Layman Lab, which was looking at this first question. So what compounds are making up the soil organic matter? So my work is really getting into the second question of where are these compounds coming from? Who is making these compounds that make up the soil organic matter? So recent research into this has looked at how microbial necromass can be contributing to soil organic matter, also called soil organic carbon. So what this study did is they got metadata from a bunch of different studies that measured microbial necromass markers in the extracted single organic carbon, and then estimated how much of a contribution the necromass was making to the overall soil organic carbon. And so they looked at this across the range of soil depths, and then actually up at the top you'll see all of the soil depths kind of smushed together. And they looked at different types of soils, so cropland soils, grassland soils, and then also forest soils. And across all these systems, as you can see from the bar graph, particularly the one up at the top, microbial necromass was measured and estimated to make up around 50% of the soil organic carbon. And so particularly focusing on this term of necromass, it's showing the importance of understanding the microbial contributions to this, the importance of microbial growth and death cycles, understanding them. So understanding how they're making their necromass and how much they're making it. So now the question becomes, if this is the piece that we need to understand, how are we going to measure growth and death? It's not a trivial thing to do. So typically, if you think about a microbe and you think about typical microbiological methods, in order to measure growth, we would use cultivation. So you would get an isolate of the microbe and grow it up in a lab culture and use that to measure the growth and death cycles of the microbe. The problem comes into soils. We are notoriously bad at cultivating soil organisms. And in fact, there was a recent study where they took a soil sample and they did sequencing to see how many different types of microbes were there. When they did that, they found 31 phyla were present in the soil sample. And then, and this is from 2019, they had this new cultivation method they wanted to use for soil microbes. And they cultivated the microbes and sequenced the ones they were able to cultivate. And when they did that, they found they cultivated representatives with 4 phyla. So cultivation is not a method that we're able to use to accurately represent the diversity of growth that could be happening on the soil system. And so because of this problem with cultivation, the field has largely moved towards sequencing processes. So instead of relying on cultivating these microbes, we can extract their DNA and sequence it. And particularly recently, do metagenome sequences where we're getting the entire genome. But coming back to this idea of wanting to understand growth and death, the genome isn't enough for us to be able to tell growth and death of work. So you can imagine perhaps didn't know what a dandelion's growth and death cycle was. And we wanted to understand how it might be different in its growth and death from an oak tree. But all you have is the genome sequence. You're not able to measure its growth and death. There's no particular gene in the genome that controls the growth and death. In fact, what plant breeders have found is there's a variety of genes. Growth and death is something that arises from the entire genome working together to create what an organism is able to do. It's not a specific pathway. And so from the microbes in the soil that we're able to measure the growth of, we know that there's differences in growth and death cycles of microbes. So this is showing the abundance of microbes over time, a growth curve for soil microorganisms. And as you can see, the blue and yellow ones have this really rapid growth and rapid amount of death, whereas the red ones have very slow growth and death. So similar to dandelion and oak tree, there are big differences in how these microbes are growing in the soil and appear to have different patterns of growth and death. But if all we have from them is their metagenome sequence or genome sequence, we can't tell from that what growth and death the microbe might be having. So we can't really understand the necromass they might be making and contributing to the carbon in the soil. And so again, we just run into these problems. We can't cultivate microbes, so we can't measure growth and death that way. Even if we have the genome sequence or we understand the phylogeny of the microbe, we can't really use that to tell us how the microbe is growing and dying, what its growth and death cycles are like. And so we come back to this issue of how do we make sense and measure microbial growth and death. So the way that the field recently has been trying to get around this is using something known as life history strategies. And so this is a term that's largely borrowed from plant ecology, which at one point ran into a similar problem. And it's based on this idea that an organism can't be the best at everything. You have to make trade-offs in your general approach to life. And then specifically trade-offs in things such as your growth, your resource acquisition, and your survival. So there's different specialties that you can feed into, or you can be a generalist in some of these specialties, and that's how you function and survive in your ecosystem. So a classic example of this is Grimes' competitor, stress tolerator, and mineral theory. And so this is a way that he used to categorize the life history strategies of different plants. So for example, a competitor, you can think of an organism like an oak tree. So this is an organism that grows slower, has more investment into the ecosystem around it. It's really good at competing for resources. Whereas you can think of a mineral or a weedy species, something like a dandelion, it grows quickly and dies quickly. Its goal is to produce a lot of offspring, and it's not investing in the ecosystem. And then finally, you have a succulent, or something that's stress or scarcity tolerant. And the goal of these organisms is not to die. They can live in stressful environments that other organisms may not be able to live in. They're not trying to grow really quickly or anything. They're just trying not to die. And so when we look and go back and think about applying this to microbes, again, we have this observation that growth and death cycles are differing in different microbial communities. So we have this kind of sense that there might be a difference in life history strategy between different microbes that could help us understand what function they're having in the soil. And so the question becomes, is there a way similar to what's been done in plants that we're able to find trade-off traits in microorganisms that we can use to define what specialty they might be having in the soil? So their investment into things such as growth, resource acquisition, and survival. And so that's really where the first question and the first part of the project comes in. is, can we build a life history framework that explains microbial growth and death behaviors and explains the different specialties and life strategies that microbes might be having in the soil? And so previous work in this area has largely started trying to apply life history frameworks to microorganisms. And one of the first questions that you have to ask yourself is, well, what traits are we trying to look at in microbes to be able to measure this? So in plants, the way, again, these concepts is borrowed from plant ecology, people have found things such as leaf area, leaf dry matter content, and specific leaf area can be related to life history strategy. And so measuring these traits tells you something about how the plant is investing in energy to be a specialist in surviving in its work and in its environment. So from this, you are able to understand the life history strategy. And comes to microbes, though, there's no leaves that we can measure. In fact, going through and looking at microbes, even through a microscope, is really an impossible task when you're looking at a soil that can have billions and billions of microbes in it. So, as I mentioned before, our way of kind of getting at and looking at the microbial community is through these genome and metagenome sequences. But again, with growth and death, you can't look at just a single gene or a single set of genes. And so instead, what we're interested in looking at is what I'm calling general genomic features. So in this case, kind of explaining what I mean by general genomic features, I'm looking at zooming out a bit on your genome and understanding what general tools do you have to accomplish different tasks in life. So an example could be a membrane transporter, or how much of your genome is made up of membrane transporters. So how much are you able to pull stuff in across your cell wall or push stuff out across your cell wall? And how might that be related then to what you're doing in the ecosystem, your overall approach to life? Another example could be transcription factors. So in the genome, transcription factors help you turn off and turn on genes. They help you control your gene expression. So if you could imagine an environment that perhaps is not good for microbial growth, it's nutrient deficient. And if we have two microbes, one that has many transcription factors and one that only has two transcription factors living in this environment. And then suddenly there's a wash of nutrients in the environment you want is very good for growth. You can imagine that perhaps the microbe with many transcription factors is able to respond more quickly to this changed environment and change its gene expression to turn on its growth and reproductive gene. And so it's quickly able to respond and begin replicating in this environment. Whereas perhaps a microbe with only a few transcription factors can't change as quickly, so it's not able to start replicating it as quickly. And so these perhaps general features such as transcription factors have an influence on the growth and death and responsiveness of a microorganism in the soil environment and its overall life history strategy. And so current work in this field has really looked at making an overall life history, first going in and defining what they think the different life history groupings and microbes could be. So using the understanding of both physiology and trying to come up with, on the first hand, what they think the groupings could be. And then they go and look for genomic features that are interesting and potentially related to these categories. So what I'm proposing is to do the opposite of that and start by looking into the genomic features, and particularly ones that seem interesting and like they would be trading off to define your general approach to life. So instead of applying the framework to the genomic data, I'm using the genomic data to inform my pre-machine. And then once I am finding and analyzing those genomic features, using those to tell me what specialties do I think microbes in the soil environment might be trading off against, and using that to define my life history groupings. And again, so we have evidence. I've shown this figure quite a few times. Microbes are growing differently in the soil. They have different growth and death cycles. We also have evidence that general genomic features, like I mentioned, so in this case, membrane transporters whether the transcription factors are associated with each other. So in this case, you have an organism, these two factors are positively correlated with each other. We have high membrane transporters who also appear to have high transcription factors. And so we can imagine an organism that occupies this side of the graph might be having a very different life history strategy than an organism that occupies a point where they have low transcription factors and low membrane transporters. And so that really defines what the first part of my work aims to build on, is examining genomes and metagenomes from the soil and looking for these general genomic features that appear to be showing a trade-off indicative of specializing into a certain life history strategy or have interesting relationships to the potential growth and death or life history of an organism. And then from that, I would use those features I've identified to understand what those different specialties could be and how those could be related, again, to how the organism overall is approaching its life history and what it's doing in a soil environment. And so at the conclusion of this, my hope would be that we could take the genomes of organisms and the metagenomes of organisms and use that along a plot like this to understand what sort of niche and what sort of ecological niche the organism is occupying. And from that, get a sense of its growth and death, as well as other factors related to how it's functioning. And so when we're thinking back to this growth and death, we want to also understand why would this be important for soil carbon. And so we have some initial hypotheses about how some of these potential groupings could relate to soil carbon. So if we had a growth adapted organism, we think about a rooterole or a dandelion. Perhaps that's an organism that's producing large amounts of liquid mass. It's growing and dying rapidly in the environment, so it's producing a lot of mass. And then if we go to a competition adapted organism, we think about like an oak tree. Perhaps it's producing an intermediate amount of liquid mass. It's not growing as much, it's not dying as much. We're still trying to understand how that can relate to this production. But then if you think about a scarcity adapted organism that's goal is just don't die. It's not producing a lot of liquid mass. And you can imagine how being occupying these different spectrums of specialty that then go on and influence how the microbial community is contributing to soil carbon and the accumulation of soil organic matter. And so that is the first part of my work, is kind of nailing down and defining what these different life history strategies could be and how we could achieve them. And the subsequent questions I would have is maybe going back into the field and getting closer and closer back to that question of the influence on soil organic matter. So one of the questions would be, do our management practices influence what life history groups are present in the soil, or what the relative composition of those different life history groups are? So for example, I would look at tillage, because we know tillage alters the composition of the microbial community, but we don't know how it might impact what ecological niche these different soil organisms are occupying, or what their specific specialties might be. And that could be what's important for understanding microbes and soil carbon. And then the final question, which is really related to that, as I mentioned, do these different life history groupings actually matter for soil carbon? So we have some hypotheses, again, about how they could be related to macromass production, but actually going out in the field and using soils to study if these life history strategies, differences in that composition, have consequences for the eventual carbon role in the ecosystem. So that really is a summary of what I'm going to be doing, focused heavily on my beginning work and then alluding to what I'm doing in the future. But, thank you all so much for listening, and I'll be happy to take any questions.
The nature of genes, cells, but virus in the spectrum, but how genes are completely told microbial.
Right. Well, that's honestly is a big problem for understanding genomics. I think that using the life history strategy that also actually allows you a little more breadth to take into account these effects. So if they're swapping genes that would be important for changing your life history strategy, I'd be setting up a framework that would take that into account. So I think the swapping of genes really becomes an issue if you're trying to focus specifically on phylogeny and understanding how phylogeny, if you these two species are closely related, but this one picks up a bunch of genes that are not, it didn't get from the species, but history framework would be taking a step away from phylogeny and saying, what is the set of genes that you currently have, whether you got them from your ancestor or you got them from somewhere else, and what does that mean for how you're potentially approaching life or whatever your strategy is.
So this might be more indicative of when this narrative changes versus what's actually going on. Yeah, so what's going on right now, like history works, file comes through, change. So yeah, I get it. Thank you.
Right. Yeah.
A very naive question. How do you measure necromancy? I can see a dead animal or a dead plant.
Yes.
How do I know that that thing in the soil is a dead microbe? You still see the membrane or what?
So what they use is a marker of microbial cell wall composition. So in that study specifically, they broke it up into bacterial and fungal. So they extracted the soil organic carbon and measured the amount of uranic acid, which is a bacterial cell wall component, and then glucosamine, which is a fungal cell wall component. And then they had an equation for converting that into an estimate of necromass based on how much of the cell wall.
That cell wall showing that it came from a microbe.
Yes. Yeah, those are unique signatures to those. Yeah.
Yeah, just to follow that question about, I think the method you kind of told the amino sugar that is kind of microbial necromass. But I mean, for the conversion factor, you mentioned that you factor earlier too. I think there's a lot of debate on that.
Yes. Yeah. So it is an estimate. But as you may have learned from the presentation last week, and as many people in this field have said, it's really hard to tell what's going into the soil organic matter and what's not outside. So those are signatures that we're trying to estimate their contributions to the soil organic matter.
Actually, I have another question about, I'm kind of very curious about your second study about, you mentioned about how the management affects the microbial discovery life, and you take change example. Do you have any hypothesis how change will affect the microbial life?
Yes, we do. So again, this is coming largely from concepts in plant ecology. So with plant ecology, we think about life history strategy. Tillage is something that encourages plants that are able to grow. So it's kind of like you come through and you mess the soil up and you kill all the plants that are there. And the first ones that are able to come back. So our initial hypothesis going into this is that a similar thing would be occurring in the microbial community. So you can think, especially if you have fungi that rely a lot on mycelial networks. If you go through and till that, those fungi are going to get destroyed. And perhaps in the wake of that, the microbes that are going to proliferate are the ones that are not competition adapted, but instead are growth adapted. They're able to grow quickly and take over kind of this ecosystem that's just been. So that's a hypothesis that we have.
Thank you for the wonderful introduction, and good afternoon, everyone. As he said, my name is Alex Yore, and today I'll be giving my M-Exam seminar presentation entitled Dynamic Crop Growth Data for Predicting Nitrogen Status of Maize. So as most or probably all of you know, nitrogen is a very critical fertilizer for any agronomic system, very particularly maize. So what happens is it's often overapplied in fields. And this is The simple reason is this is sort of a cheap insurance policy to protect against uncertainties in the season. A lot of things can happen from the time you plant, from the time you fertilize, to the time you actually harvest. So applying a little extra nitrogen is typically thought of as a reasonable practice. But there's a fine line between sort of managing yield loss and then offloading nitrogen into the environment. Because again, as most of you probably know, nitrogen is very reactive in the environment. It's very mobile, it can move around a lot. So this can cause a lot of ecological harm when it's over applied. You know, say you get a big rain after you apply, now you're washing the waterways. You're doing a lot of harm to the sort of ecosystem. There's potential human health effects. And then we also have, you know, nitrous oxide emissions. This is a major greenhouse gas, so this can be very problematic. Typically what's done, or what's very common, is uniform application of nitrogen in the field. So you pick a rate, maybe you add 20% or something like that, but you apply that same amount across the field, irrespective of where it's needed or where it's not needed as much. Even in precision systems where they have a little more control over rates and have a little more spatial control, typically this is done with like a static piece of information, like last year's yield map. And so you'll use some algorithm to derive a rate for particular areas of your field. But again, you're using this sort of single point data, whether it's an NDVI map or a yield map. So this sort of led me to this question of, can we use some kind of dynamic model to inform our nitrogen application rates at a spatial scale? And so what I mean by a dynamic model is, instead of taking a single point, like a single NDVI image, take more of a time series. And can we get some additional information from this time series leading up to the time we would fertilize to give us a better sense of what the nitrogen status is actually in the field? So to do this, we measured crop height about 2 to 3 times a week in our fields. We performed spectral measurements about once or twice a week. And then we did nitrogen status measurement of the actual plants. Then we fit a model to the whole thing to kind of try to tie it all together. And one thing I really want to point out is we did not use yield data in this, because I imagine somebody would ask, because any agronomic pursuit, sort of the paramount becomes yield. You know, you want to know, how's this going to affect my yield? Is it going to hurt my yield? Is it going to help my yield? Is it going to maximize it? And so I just want to point out now that we didn't look at yield in this part of the study, and this is actually part of a broader body of work that we actually do have the entire growth rate for the entire season, and we'll have yield data. But for the purposes of this project, we're not looking at that. So this took place over a few different sites. So here in Ithaca, we had 3 different sites across 3 years, 2021 through 2023. And what we did is we had 2 separate planting dates per field. So we're calling those a different site year. Because you essentially have created a different environment. If you plant 2 weeks later, you're sort of changing the environment a little bit, at least in terms of weather patterns and things like that. So we have 3 sites, 3 years here in Ithaca. We also partnered with our colleagues at Agriculture and Agriculture Canada, which, if you're not aware, is essentially the Canadian version of the USDA. They provided us with 1 year of data this past year, 2023. Same protocol where they did 2 different planting dates, and they had 3 different fields. They're calling them blocks, but they're 3 separate fields within this area. So this is a map of theirs at Buckley, which is just south of Montreal. So there's their 3 different fields. Again, 2 planting dates, as I mentioned. The little squares we'll talk about in a moment. These were our 3 sites. So 2021, we were at McGowan Woods. 2022, we were at Caldwell Field, which you probably know is over by the greenhouses. And then 2023, we were a couple miles away in Varna. Most of the rest of the talk will focus primarily on 2023 data, with a little bit of 2021 and 2022, primarily on last year's data. So what we did is we mapped out the field. As you can see, Professor Longchamps here with his backpack with this funny little antenna on it. So what this is it's an RTK GPS. And what this does is it allows us to have sort of centimeter level resolution within the field. And so we can walk around the field and know where we are within a couple centimeters accuracy, or within about a centimeter accuracy. And what we did is, so as promised, the little squares. So what these little squares are, they're 3-meter pixels within the field. And so we flagged out the corners of these. And again, that's why we needed this high resolution GPS. so that we could map this out precisely. The reason we chose the size and the orientation is because this is based on planet satellites, which, if you're not aware, is sort of it's a daily pass, high resolution satellite product. Basically, they've sent an entire swarm of little satellites up into the sky, and so it allows them to give us daily imagery. So we matched that to their grid so that we can use this for further work with the satellite data. And as I mentioned, we have two different planting dates. So the southern half of the field was planted around late May, the second half of the field was about 2 weeks later. For the height measurement, pretty straightforward. You can see up there, there's sort of a zip tie encircling the plant. So we did this with all of the plants. We actually went back and measured the same plants every time we measured. And there are 10 of them within each field with a specific colored zip tie. So we can actually track the growth of individual plants. This doesn't play as much into this particular project, but we did want to have that granularity for later. Ultimately, what we did is we averaged the heights. So we have sort of a average height for the individual 3 by 3 meter pixel. This corresponded well with what they did in Canada, which is where they just averaged the average value, didn't have this sort of granularity of data. We measured from the soil surface to the highest leaf. And I want to point out that we did unmanipulated. That is, we didn't move the leaf in any way, or we didn't stretch it out. So there's a pretty standard protocol. A lot of times you will, hold the leaf up to its highest point and measure that way. The reason we chose not to do that and to do it this way is we wanted to correspond this more closely to what you would see with, say, a LiDAR image, where you're not actually touching the plant, you can't affect it. It's just how the plant sits, where it is, what is the height that way. So we wanted to measure that way. And as I said, there were 10 plants per pixel that we were measuring, and we also averaged those for an average height over that 9 square meter area. There we go. So for the spectral measurement, we used the ASD field spec handheld. Although we didn't use it handheld, we used it on this boom and on a ladder. So we did this to get about 4 meters above the soil surface. The reason we did this is because the spot size of the actual device comes out to about 3.5 meters at that height. So it worked out perfectly, nearly perfectly with our pixel size. So we would stand up on a ladder and measure straight down. That was how we got our spectral data. One of the challenges with this is it requires a lot of calibration. So you can see on the right, my colleague here holding up this device called Spectralon, essentially just like a pure white reflective surface that we were able to get sort of a white balance for our device. But we had to do this every 10 to 15 minutes in the field. So as we're going along, we're constantly recalibrating to make sure we have the most accurate data. Now, our wise Canadian friends used a drone to get their spectral data. And having walked through the field with a ladder a lot, I'm a little jealous. So again, because we were trying to tie this to this sort of planet satellite data, what we did is we took this, so this figure on the right is sort of a typical day's measurement from the ASD. And it's a hyperspectral instrument, so we're actually getting single nanometer resolution from 375 to 375 to 1025. And what we did is we broke this down into individual bands. And these bands correspond to the planet SuperDove, which is the current instruments that are up right now. So we wanted to match up that band structure so that we could ultimately use this data and use this protocol with the satellite data later. So for nitrogen status measurement, we did in 2021 and 2, we did a nitrate sap test. So this is taking fresh plants and actually measure the amount of nitrate within that. Again, since we're not focusing as much on the 2021 and 2 data, don't worry too much about that. So for 2023, what we did is a nitrogen nutrition index. What this is essentially a measure of the nitrogen in the field or the nitrogen in your plants relative to a sort of critical nitrogen concentration. This critical nitrogen concentration is a sufficient level for growth, at least theoretically. And it tends to be a pretty good standard, pretty good hallmark for this kind of work. So for this, we actually took dried plants. So we did destructive sampling in and around the pixels in question, and then derived our sort of NNI values from that. This protocol is also followed in Canada, so we have consistent measurements for nitrogen. So this is a typical plot of our height. So the individual points are the actual measurements where we measured the height. And what we did is we fit this exponential curve. And so if you know a little bit about sort of corn growth or even plant growth in general, if you look throughout if you sort of project this out through the whole season, you're going to see sort of a logistic curve tends to fit really well. Because we're not looking at the entire season, we're just looking at this early stage growth. The exponential model actually fits really well. And if you've seen a logistic model, essentially the first half of it is an exponential. So this actually fit really well. From this curve, we generated a growth rate, which is essentially how fast is it growing at that last measurement date in centimeters per day. So our exponential fits were really good. Our mean R squared was 0.997. These exponential fits. I mean, you can see that there's pretty strong relationship there. It fit really well. So we went with this, calculated the growth rate from that. This is for 113 pixels. So this is including the Canadian as well as the Ithaca locations for all 3 years. We did a similar process for NDVI. Again, the points are the actual measured NDVI, or it's calculated NDVI in this case. We calculated sort of an NDVI growth rate or the NDVI increase rate. So how fast is the NDVI increasing at that last measurement day right before side dress? The fit was not quite as strong for exponential, and you can just see that, this is just a typical plot, and the points were a little bit off. But our mean R squared is about 0.86. The N is a little bit lower, so we had only 95 points here because the 2021 data would not have spectral data. So that's not included in this. Now, looking at our static model, and again, by static model, I'm saying a single data point. So if you're just looking at this one point, just the height, you measure one time right before side dress, you measure the height. How good of an indicator is that of the nitrogen status? Not bad. I mean, we're looking at about a 0.68 R squared for this. This is NNI versus height. But what we wanted to see is, okay, we're looking specifically at this one day right before side dress. What about all the other days before that? Are there other days that might be a better predictor? And so as we sort of move down along this curve, looking towards planting date, we see the R squared drop off pretty precipitously. So there's no real improvement here with the static model for looking backwards in time. We're trying to predict it a week earlier. Now, if we look at our dynamic model, again, this is height. So we're looking at NNI, but now we're looking at growth rate. So this is the slope of that curve essentially at that last point. So this is our growth rate, the dynamic model, which we've derived from the actual exponential curve. So for this, our R squared is about 0.64. Still not bad for predicting nitrogen. And again, we wanted to look and see, okay, what if we moved back through time and sort of move our way through here? How good of a fit do we get? And you see there's a little bump. So the red line indicates the R squared of our left-hand plot, which is measuring just at the last date. But now if you measure about a week earlier, you get a pretty significant bump in accuracy. So we're actually just a little bit higher than the static model in this case, about a week earlier. So this is actually good news because typically you want a little bit of lead time to be able to plan this thing out. If you're going to use this sort of tool to indicate how much to fertilize, it'd be nice to know that a few days earlier. Also, sort of projecting forward into using satellite data, this would be really important because, as we all know here, clouds can be a big issue with satellite data. So having that window of time where you actually have still a decent accuracy is really important. So now again, looking at the static model, this is for NDVI now. Same thing, if we just take the NDVI value at that last point, what do we get? The R squared is not great, about 0.3, 0.33. Again, if we look and project this out and look back in time, how early can we get a better measurement? Well, we really can't. Anytime you go back through, it's going to just drop. One thing I do want to point out, though, is for our height model, the actual measured height and the projected height for our last day were spot on. They were the same. The model and the measured were almost identical. You saw that from the 0.99 R squared. In this case, there was a little bit of a discrepancy between the modeled values. So the red line here is the actual measurement R squared, so corresponding to the figure on the left. But now this is looking at the point, the actual measurement. But if you take the point on the curve as opposed to the measured point, we actually get a little bit better accuracy that way. But again, moving down, going back through time, our accuracy just drops if you go earlier and earlier in the season. Now with our dynamic model, for NDVI. And again, this is looking at that increased rate of NDVI. How fast is the NDVI increasing at that last date? In this case, we got an R-squared of about 0.49. So this is a pretty significant improvement over the static model. And so this sort of underlies this sense of, using a single point NDVI to try and predict nitrogen status is maybe not the best, and a dynamic model may actually have some improvement over this. And similarly, as we go back in time about 5 days, we get a little bit of a bump and a little bit of an increase in the overall accuracy of the model. Now, comparing the static and dynamic models, as we just mentioned, the overall R-squared was actually just a little bit higher for the static model, but this is only for the date right before side dress. If we actually look across all days, again, we have this little bit of room for improvement, which takes the dynamic model, just kind of ekes out the static model in this case. But again, because now we're a week earlier that we can make this prediction, that's really important, one, for planning purposes, and also giving us a window as we sort of move this process into satellite data. Similarly for the NDVI, except in this case, we did see a pretty big increase between the static and dynamic model directly at the last day. But then we also have this little bump of improvement about 5 days later. Again, this is really helpful for planning and also sort of mitigating issues of trying to get satellite data right on the date that we need. So to sort of wrap this all up and bring it together, so we looked at these dynamic model fits. So our height model, there was not really any improvement with the dynamic model over the static model at the final measurement date, but we did see a slight improvement about a week earlier. So if we're predicting a week before side dress, we actually have a slightly better chance of predicting that nitrogen status. With the NDVI, we saw about a 50% improvement in the accuracy of the dynamic model versus the static model. We didn't get quite as much of a benefit from going earlier, but there was still a benefit, and the overall fit was better. Some of the limitations of how this study went, one of the ones is just the noisy spectral data. It's just inherently noisy. I mean, you saw how we're doing it. I'm up here on a boom. We were pretty accurate about our heights and things like that and aiming, but there's just inherent challenges with that. A tiny cloud passing in front of the sun when you're taking the measurement can really affect that spectral reading. We did average it over multiple measurements to sort of take care of that as best we could, but there's still a lot of noise in there. One thing that we didn't look at that I think would be really helpful is looking at the differences between site years. And this is site years from one half of the field to the other with the different planting dates, but also between the different sites, that is Ithaca versus Canada, even looking at our previous years and seeing the differences there. There are inherently going to be differences in precipitation, the amount of GDP that they're starting with in the first couple of weeks to make a really big difference on the growth. So that's something that needs to be looked at a little more deeply. And again, as I mentioned in the beginning, we are not looking at yield. And again, because this is sort of a paramount issue for any agronomic experiment, ultimately somebody wants to know, a farmer wants to know, how does this affect my bottom line? How does this affect my yield? It's going to hurt my yield. It's just going to be a waste of time. So, as we use the broader body of work that we have, the broader body of data that we gathered from this project, which actually looks at the entire season, then we can start teasing out the yield impacts and things like that. So the implications for future work, as I mentioned, we want to look at using satellite data to do this. As fun as it was hauling a ladder through 7-foot tall corn, I would love it if I didn't have to do that. And I can tell you, I don't know many farmers who would be really willing or even have the time to do that. So, drones are great if you have access to one. That could be a really great way to sort of gather some of this data. But ultimately, it would be having this public access satellite data would be really powerful for sort of making this predictive. Ultimately, what we'd like to do is make these high resolution prescriptive end maps. So you could map out your field at 3-meter resolution and know where needs less nitrogen, where is going to need more, just based on this sort of predicted nitrogen status using NNIs, as I mentioned before. So with that, I'd like to thank you all for being here. A big thanks to my advisors, Professor Longchamps, Professor Van Ness, as well as Bill Philpott, who's helped a lot with the spectral data, the entire team at Agriculture and Agriculture Canada, The entire Longchamps lab, past and present, they've all been a part of this project and have been tremendously helpful, as well as the Cornell Institute for Digital Agriculture for providing some funding for this project, and then Cornell Farm Services for all their awesome help in getting our fields prepped and helping with all the sort of management of the actual agronomy of the thing. And of course, I'd like to thank all of you for being here, and thanks for listening.
So, you mentioned Harold at the very end, and so I was already thinking to ask you how this might integrate with the adapt-in approach, which for that side dress date, we'll give, because of having followed the weather, the initial applications and so forth, it's going to give some estimate of the nitrogen status in the soil at that point. You see a complementarity to this I mean, you're being more spatially resolved, obviously, that's one aspect, but have you thought of some integration with adapt-in?
Yeah, this is something we talked about a little bit. This could essentially be another piece of that, another way to enhance the sort of reliability of that or enhance the accuracy of the sort of predictions that's going to make. I think this could be a really important piece of that, taking some of that dynamic data as opposed to just a single NDVI map. I think that could be helpful in that context.
Thanks for your presentation. So as far as I grasp, the idea is basically that we use growth rate as an indicator for nitrogen status because nitrogen status is probably what determines our growth. So have you thought about other factors that can be influenced the growth rate at that point, like moisture status, maybe some limitations, sunlight that can affect your measurements of your estimation?
Absolutely. There are, because there, like I said, especially between the different site years, we're going to have differences in the amount of GDP that's accumulated early in the season, moisture differences, soil differences, weeds can be a big factor. So that, yeah, there are a number of other factors that I think should be explored to really make this a more robust model.
Nice presentation. So, if you use the plant height as a parameter to characterize the model, so you are aware of short scatter point market, how is the cost translate to those point height?
That's actually a really interesting question. So we used, the only difference between varieties was between Canada and us. All three of our years we used the same variety of corn. So I think that'd actually be a really great thing to look at and see, does a variety that typically is going to be shorter, do we see the same thing? I think that'd be a really great thing.
Maybe like we didn't see much difference between the last height and the growth rate for the measurement. But in this case, maybe the growth rate was more stable across varieties than the actual height if you have some shorter, taller. All right, thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.